All right, so as always, guys, okay, you'll see in Google Classroom here, okay, under the class work tab, you'll have the PDF document, which is the lab sheet with all of the text and questions and stuff on it, and then there's your version of it that you have to fill in, okay, the Google Doc. That's what you should have open right now. Actually, you probably have both open, but you'll need to be filling in some stuff, notes to self, okay, for the pre-lab stuff here for the cell anatomy lab. Okay, the purpose of the cell anatomy lab is that we want to find out what parts of a cell are common, okay, in many different cells, and as a result are the most important or are carrying out important or let's say basic functions. All right. So the problem we're investigating here is cells must carry out the basic functions of an organism in order to survive. Point number 2 of the cell theory. Okay. Thus, they must have structures within their cells that carry out those basic functions. All right. So the lab is going to work like this. We're going to look at a plant cell. We're going to look at a generalized animal cell. So the plant cell we'll be looking at is from the leaf of like any normal plant, okay, like any normal kind of house plant. So we'll be looking at a plant cell from there, and then we'll be looking at an animal cell that is actually from tissue that is called stratified squamous epithelium, which is the really complex way of saying, you know, when you dust your knuckle and there's that red skin that really stings underneath, that stuff. Okay, we're taking a sample. Well, we won't be taking a sample. There will be a sample under glass for you to look at from there. Okay, so we'll be looking at that as our generalized animal cell, and then we are going to look at one of your cells. Okay, so it will be another animal cell, obviously. The way we're going to do that is you're going to take a swab of the inside of your cheek with a Q-tip. Okay, so it's very simple. Just take the Q-tip and kind of put it against your cheek and twist it and move it around a little bit and then smear it on the slide. Okay, then you put a drop of water, just like you did with the letter E, and make a wet mount and look at it underneath the microscope. That one will be a little tougher to find because it's not stained. Okay, the other slides you're going to look at will have stains added to them, so it'll be color. Okay, this one will not, but you will be able to find your cells on there as well. If you're looking at something that's really dark and opaque, it's not a cell, it's a piece of food. Okay, if it's a cell, it'll be quite a bit more transparent. Okay, and if you're having trouble telling, I'll be able to come around and help you. All right, but those are the 3 cells we're going to be looking at. Okay, I'll also have a prepared slide of human blood, but it's not part of the lab. People just always ask, can we look at our blood? No, you can't look at your blood. Okay, yeah, no bodily fluids, we're not allowed. Okay, infectious diseases, hepatitis, that kind of thing. So no, don't prick your finger and look at it under the microscope, please. Okay, I don't want that. So I have a prepared slide of human blood if you want to look at it. But with the main 3 that are important are the plant cell, the animal cell, and your cheek. Okay, so these are all cells from eukaryotic organisms. That is, organisms that have compartmentalization, we talked about that yesterday, in their cells. All right, so we're going to be looking at those and trying to find out what kind of organelles do they have in common, what are some differences, okay, why are those differences present, stuff we talked about in the modes of nutrition lesson, things like that. Okay, so in the design here, manipulated, responding, controlled variables, okay, what are you looking at under the microscope? Each time you look, it's going to be, yeah, from different organisms, right? We got plants, we got animals, okay? So it's important that we look at a wide variety of cells, okay? The responding variable has to do with what we're looking for in those cells, okay? Write it differently than that, probably be more specific, but that's as specific as I'm going to be, okay? Talk about that. Controlled variables, okay? One of the most important controlled variables is that all of these cells come from eukaryotic organisms. Okay? It would hardly be fair to be looking for cell structures if one of the organisms was a prokaryote that doesn't have them. Right? We talked about eukaryotes, prokaryotes briefly the other day. Okay? That's one of the controlled variables. Okay? You guys need to think of 2 more. Okay? Things that are the same each time you look at a different cell. Okay? What are some other things that are the same? Okay, for our hypothesis, we want to generate a hypothesis about which organelles will be present in cells and visible under our microscopes. That's probably more to do with the then part. Okay, the if part probably has more to do with this. Okay, with the problem, with the cell theory. Okay, not with the problem that there's no problem with the cell theory. The problem that has the cell theory in it. Okay, so the if part should probably talk about that. okay, and about what cells have to do. Okay, the and part, as always, describes the experiment. Okay, what are we looking at? Where are they from? That kind of thing. And the then part needs to be a specific prediction of what organelles you will see. And probably a brief blurb about why. Okay? Keeping in mind that while every cell probably has ribosomes, are you going to see them through a light microscope? Nope. Okay, remember, what are the ones you're going to see? You can maybe mention that ribosomes are in every cell, but you're not going to see them. Okay, they're way too small. They weren't even discovered till the electron microscope came about. Right, so they're not something we're going to see with the light microscope. Okay. All right, questions on the hypothesis? Okay, remember if, and then, okay, we got to have those things in there. All right, so the procedure, like we said, get your microscope, get the eyedropper, set the microscope up, okay, one slide at a time, same as the last lab back at your desk. It says here toothpick, but I forgot to change it. It should say Q-tip. Okay, I had too many people drawing blood using a toothpick to get their cells. I know it's stupid, but people were like, one kid, I thought he was going to poke it right through. It's going to come out the other side of his cheek. Okay, so yeah, just a Q-tip. Okay, it works for CSI. It'll work for us. Okay, collects enough cells. So the order you should probably look at these in would be look at the monocot dicot cross section. That's the plant. Okay, then you're going to look at the generalized animal cell. Then look at your cheek cell, and then if there's time, you can look at the human blood smear. Again, the human blood smear, you're not going to do a lab diagram of that. It has no part in the analysis. Okay, so it's just for kicks. Okay, just to look at. All right. All right, then for our analysis here, there's only one analysis question, guys. That said, I'm going to want a lot of information here. Okay, probably a couple of short paragraphs for this single analysis question. Okay, based on your knowledge of cell structure and your observations of the cells in this lab, what organelles are most important for a cell to function? Okay, and which ones are specific to a certain mode of nutrition? Explain your reasoning. So probably one paragraph would be about what organelles are most important, why, okay, did you see them, where did you see them, okay, that kind of thing. And then another one about, well, in this one I saw this organelle, but it wasn't in the other cell. Maybe that has to do with it. This one is this kind of an organism, this one's this kind of an organism. Okay, so it's kind of two separate parts to that single analysis question. Okay, if you write, the nucleus is in all cells, so it must be important, and leave it at that. That's definitely not going to get you 5 marks. If you're lucky, it might get you 1 out of 5. Remember, this single question is 5 marks. Okay. All right, conclusion, copy paste your hypothesis, and then looking at your results, what did you see? Okay, explain if you can accept or reject it. Sources of error, okay, you know, think about 2 sources of error and ways to fix them. Remember, they can't be human error. All right, the stuff you'll be looking at tomorrow, okay? So this one here in the top left, okay, this is the plant cell, right? There are actually a number of plant cells in this picture, right? But basically, this is the top layer of the leaf up here, okay? So this layer up here is called the epidermis. It's the layer that's like skin, and it secretes the waxy part of the leaf. Okay? Underneath that is what we call the palisade layer. That's the layer we want to draw the cells in. Okay, so for example, this here is a plant cell. Okay, that's the nucleus. Okay, all these little grayish yellow kind of things are chloroplasts. Okay, if you look closely at the bottom of the nucleus right here, okay, you can see it's slightly different than the top of the nucleus. That's because that's the rough ER. Okay, I'm not saying use your imagination when you're looking at this, but use your imagination a little bit when you're looking at this. Okay, you know, you can see quite a bit of stuff. All right, this whole area here that looks completely clear is likely where the water vacuole is. Okay, in this particular cell, the water vacuole is behind the nucleus and the chloroplasts, whereas on this cell, the water vacuole is in front of them, so we can't see them. Okay, we can't focus on them, or it's not focused on them right now. Okay, everybody kind of follow me on that? Now, when you do your lab diagram, okay, obviously you're going to have more than one cell in there, right? Do your best to do your labeling from one cell, but if you have to do your labeling, if you have to label something in another cell, that's fine. Okay, does everyone follow me there? But that's what we'll be looking at. Okay, you look at the different layers, and we'll actually talk about the different layers of the leaf later on in the unit. So this is what we call the Palisade layer. That's where all the cells are rectangular and look a lot like the diagram you have. And then down here is the spongy layer, and all the cells are very irregular, okay, irregularly shaped down in that layer. But you can still see, a nucleus and chloroplasts and stuff like that. So, okay, so just so you have an idea of what you're looking at tomorrow. All right, so that's the plant cell, okay, and then we have this here is the stratified squamous epithelium. All right, this is the animal cell. Many, many animal cells. All right, so see this purple circle here and this purple circle there? Those are nuclei. All right, so to give you an idea here, this is one cell, okay? That's its nucleus, okay? And if you look closely at a lot of these nuclei, you see kind of on one side, they're kind of fuzzy looking. What would that be? That's the rough ER, okay? If you can see the outside of the cell, that's the cell membrane, right? You can label that. Everyone kind of following what I'm looking for there? Again, you're not really using your imagination, but you're using your imagination, right? So it won't be this big when you look at it under the microscope. Okay, it'll be smaller than that, but just so that you get some idea of what to look for. Okay, if you get one that's dyed a little bit differently, it may look more like this one. Okay, and again, each one of these is an animal cell. There's a nucleus. There's a nucleus. Okay, there's lots of them in there. Okay. All right. Then you'll be looking at your own cells, okay, from your cheek. These are mine. Okay, so here's the nucleus of my cheek cell. Okay, this is my cheek cell right here. Okay, you can see all kinds of different parts of it. Okay, you can do a pretty good job. But as you can see, it's not dyed. All right. The way to get it to stand out is to use that little condenser diaphragm on the bottom of the microscope. Okay, as you adjust that, it'll change the contrast and they'll just jump out. Okay. This is a different slide of the same thing. Okay, again, it's just some of my cheek cells. Right, that's what they should look like. If it's big and green or dark, it's a piece of food. No matter how well you wash your mouth and brush your teeth, you're still going to have pieces of food in there when you do this lab. Okay. All right, and then the blood looks like that, but again, the blood's not really going to be all that important for the lab. Okay, those are the things we'll be looking at tomorrow. All right, we got a few minutes left here, about 8 or 9 minutes. You can work on your pre-lab stuff, okay, work on your design hypothesis and things like that. So tomorrow we'll come in here and right away we'll do attendance and we'll go across the hall and start working on the lab. So you'll have the whole class to make sure you do a good job of finding everything, getting good pictures of the cells and all of that.